Hey everybody, welcome back to the last week in April, and we are going to move forward, and we have just finished up a unit on how cells make energy with cellular respiration. And that is, the overall equation is represented by C6, H12O6, which is glucose, is broken down, and then way at the end, what is left of glucose combines with a bunch of oxygen and spits out we spit out 6 CO2 a while back, and 6 H2O, 6 water molecules. All right, so we've seen this 100 times before, and you already had an exam on it. So this is done in the mitochondria. Oh yeah, and we get 36 to 38 ATP out of that from 1 molecule of glucose. We calculated all that. For the last part of our unit on how cells make energy, we're going to look in another way that a certain cell type does make energy, and for that, we need to go green, because what I am talking about here is photosynthesis. I misspelled synthesis. Photosynthesis. Hard to talk and write at the same time. Photosynthesis occurs in the chloroplast. The chloroplasts are an organelle in plants and algae, and some bacteria, and not really, and cyanobacteria. They're special. It's more like cyanobacteria actually are a chloroplast. That's just a little piece of trivia there. I'm not going to test you on that specifically, but if someone is watching this going, wait a minute, cyanobacteria don't have chloroplasts because they don't have organelles because they're prokaryotic, but cyanobacteria basically function as a living chloroplast. But chloroplasts otherwise are organelles in plants and algae. And I'm pointing this out because algae are not plants at all. They lack the complexity of a plant body. They're not plants, but they are photosynthetic. So for example, phytoplankton are a type of algae. And they are unicellular eukaryotes, and they do photosynthesis. Phytoplankton live off of, I don't know, some sort of high percentage of the world's oceans. And they're doing photosynthesis over much of the world's ocean. And actually, I'm doing this lecture on Earth Day. And one of the reasons that we strive so hard to keep the world's oceans clean is that all the garbage and crap that we pollute the oceans with actually kills off the phytoplankton, which means they cannot do much photosynthesis. And that is a big, hairy deal, and here's why. The overall chemical reaction for photosynthesis looks like this. Oops, come back, green. 6 molecules of CO2, and this doesn't happen all at once, but eventually 6 water molecules get consumed, and in the presence of sunlight, plants can use the CO2 to make a sugar molecule, this is glucose, and spit out 6 molecules of oxygen. Does this look familiar? I sure hope so, because take a look at this back up here. Cellular respiration, let's get a different color here. Cellular respiration takes 1 glucose molecule, combines it with oxygen, and produces 6 CO2 and 6 H2O molecules, Whereas, oh, I don't think, yes, there I can get it all on the same screen. Plants take 6 CO2 molecules and 6 water molecules and make a glucose molecule and spit out a bunch of oxygen. Do these look familiar? I hope so, because photosynthesis, this overall equation down here, looks like aerobic cellular respiration backwards. It looks like this going this way, right? Let's erase this stuff because it's kind of in the way there. There we go. Yeah, so photosynthesis looks like the opposite of cellular respiration, and somehow it's still producing energy for plant cells. Now, human cells, animal cells can't do this. We don't have chloroplasts. You need chloroplasts to be able to do this. But let's take a look at what this means. Plants take the CO2, the CO2 that we are breathing out. Humans breathe this out. Humans and mammals breathe that out. Combine it with water, you're going to water plants, right? And in the presence of sunlight, they build a sugar molecule and spit out a bunch of O2 that we breathe in. We breathe this in. So what the phytoplankton up here are doing is they are basically, we say that they are recycling oxygen back into the atmosphere. They help maintain a balance. That's why the phytoplankton that are living on the surface of the world's oceans are so important, and why we don't want to continue to throw garbage into the ocean and acidify the ocean and kill off the phytoplankton. That's why the rainforests are so darn important. You've probably seen people doing fundraisers to save the rainforest. The rainforests of the world are such a high density of plant life that is doing so much photosynthesis. They're putting out a significant amount of oxygen into the atmosphere that we need to breathe in. They're actually consuming CO2, which is a greenhouse gas. They're consuming it, and they're putting out O2. So we really need plants. Humanity, the world, in order to stay in balance, needs to have plants and algae and photosynthetic bacteria even producing more oxygen, taking up all that CO2 and spitting out O2 for us. So it's really amazing how this all works. All right, I am not going to go through a gazillion details about photosynthesis for you all, so breathe a heavy sigh of relief right there. All right, but I do need to give you an overview of this, as to how photosynthesis works and what plants are actually doing here. All right, so photosynthesis is kind of like cellular respiration, yeah, only it's backwards, right? But it happens in stages. Like aerobic cellular respiration happens in glycolysis, the citric acid cycle, and electron transport, and chemiosmosis, something like that. There is no glycolysis and citric acid cycle in photosynthesis, but there are some stages. So for example, to accomplish photosynthesis, there are two main stages. Or let's say let's say stages. Stages of photosynthesis. There are there's number one, what we call the light dependent reactions. The light dependent reactions, they depend on light. Did I spell dependent correct? I can't remember. They both look wrong to me. There are the light dependent reactions, and part two, the light independent reactions. I'm going to abbreviate independent so that I can fit it in reactions on the page. So over here on the left, you have the light-dependent reactions and the light-independent reactions. All right. One of these depends on light. That would be this one here. You need sunlight for the light-dependent reactions. The light-independent reactions over here on the right do not require light. So the light-dependent reactions require sunlight. The light-independent reactions do not require sunlight. All right, so the light-dependent reactions, we used to, by the way, just call these the light reactions, and the light-independent reactions over here we used to call the dark reactions. The dark reactions. But then people thought that they had to happen in the dark, and that's not true. It's just that they don't need sunlight. It doesn't have to be dark, but the light-independent reactions can happen whether it's dark or light or whatever. They just don't need light. They don't care. So sometimes, you know, if you're out there looking or reading up on photosynthesis elsewhere, you might find somebody calling them the light reactions and the dark reactions. We don't really call them that anymore. But sometimes my mouth has a hard time wrapping around light-dependent and light-independent. So I'm going to be really careful about that. But first, the light-dependent reactions. Let's get a different color here. What the heck? Let's go with blue. The light-dependent reactions basically use sunlight to produce ATP from water. Really wild. So let's take a look at this back here. Well, no, let's not. I'll go back and look in just a sec. All right, the light-dependent reactions use sunlight to produce ATP from water. The light-independent reactions use up all the ATP from the light-dependent reactions. They use up all the ATP from the light-dependent reactions, and they use up all that ATP to basically take a bunch of CO2 molecules together to build a sugar molecule. So they use up all the ATP from the light-dependent reactions to put 6 CO2 molecules together to make C6H12O6, basically a sugar molecule. That's under ideal conditions, but they do basically make sugar molecules. Okay, so let's write out, leave yourself some space over here. Yeah. But I'm going to come down here and again write 6. I'm going to give myself a little more space. 6 CO2 plus 6 water molecules. Eventually, in the presence of sunlight, after the light-dependent reactions and the light-independent reactions, we will end up with a sugar molecule and a bunch of oxygen that we get to breathe out. Yeah, that's nice and balanced there. All right, now remember for aerobic cellular respiration, I had you connect one of the reactants to the products. Let's do that again. Like I'm looking over on this side over here, and I'm seeing that there is carbon only in CO2. Whereas over here on the product side, the only molecule that I see that has any carbon in it is glucose. So I'm going to connect those just like that. Meanwhile, I'm going to connect the two remaining ones like that. All right, now let's look up here. Right here. Carbon dioxide getting converted to a sugar molecule. Is this being oxidized or reduced? Remember, Leo goes grr. If you lose an electron or hydrogen atom, you're being oxidized. If you gain an electron or a hydrogen atom, you're reduced. So over here, is carbon dioxide gaining or losing hydrogen atoms? Oh wait, it's gaining them. So this is the reduction. This is a reduction reaction. And down here, water becoming O2. Is this an is it being oxidized or reduced? Leo goes grr. Lose an electron, it's an oxidation. Boo. Lose an electron or hydrogen atom, it's an oxidation. So this is being oxidized, isn't it? Oxidized. Okay, now if you remember when we did this for aerobic cellular respiration, back up here, where's aerobic cellular respiration? Here, let's get a different color there too. Let's go back to blue. I had you connect these two. And we're like, oh, well, glucose is losing hydrogen atoms, so it's being oxidized, yeah? And so we connected the other two. Let me clean this up just a little bit. And we were like, oh, oxygen is gaining electrons or hydrogen atoms. It's gaining hydrogen atoms to become water, so this is a reduction. But we saw through the process of glycolysis and the citric acid cycle that this oxidation happens first. And the reduction, oxygen taking on all those protons and electrons after chemiosmosis made a crap ton of ATP for the cell. This happens way the heck at the end, so it happens last in aerobic cellular respiration. Going back to photosynthesis. This part with carbon over here, carbon becoming CO2 becoming glucose, that's the reduction. That actually happens last. And it happens last in the light independent reactions. Whereas water, losing its hydrogen atoms and what's left of it becoming O2, that's an oxidation. This happens first. Happens first in the light dependent. It looks like light department. But the light dependent reactions. I need the space. Okay, remember this stuff. It's going to be on the final. And I'll talk about the final another time. No, I don't know exactly when it's going to be. I haven't gotten final word on when we're allowed to give you the final. Talk about that later. Anywho, this oxidation here happens, this part happens first in the light-dependent reactions. Alrighty. Back to green because I want to draw myself a chloroplast. Here's what's happening in the chloroplast. And remember, it's similar to the mitochondria in that it has two membranes, a nice smooth outer membrane and a really folded inner membrane. It's not all wavy like in the mitochondria, it's stacked, really. And these things are called the thylakoids. They're all connected like that. So these are thylakoids. There we go. All right. what happens is that down in these thylakoids, there are these things, these molecules called pigments. So they contain pigments, right? And a pigment, if you've ever taken an art class, I don't know if anybody here ever has, but you've probably worked with pigments. A pigment is just a molecule that imparts a color. Imparts a color. Like you can go to a paint store, maybe someday, when we get to reopen. You can go to an art supply store and buy, a tube of cyan, or a tube of ultramarine. That's that bright blue that I was using up here. I'd call that close to ultramarine. All right, pigments are just molecules that impart a color. And plants have these pigments. And an example of a pigment would be chlorophyll. And there are several chlorophylls. There are chlorophyll A's and B's, etc. And there are also these things called, and I'm going to change my color to orange for this, carotenoids. Carotenoids. 3 guesses what plant is high in carotenoid pigments. If you guessed carrots, you got it right. Okay, carrots have a lot of carotenoids. And they're just molecules that impart a color. And have you ever heard of anything like with color theory, or the like, that pigments have certain colors? We see certain colors, like, let me go back to blue. Like if I have this splooge of blue here, if this were blue paint, because to be honest, I don't know how this works on computers. Like we're virtually seeing this, I'm not actually painting this with a pigment. But if this were blue paint in real life, and I just have this little blob of blue paint, the pigment molecules in this paint, we say it's ultramarine, I wouldn't say this is perfectly ultramarine here, basically are absorbing every color except blue. Absorb every color except blue. That it's reflecting blue back to us. Light, and light takes certain colors because of wavelengths. So they absorb really wavelengths for every color except blue, and that's why it looks blue to us like that. So pigments work by absorbing light of a particular wavelength. So pigments absorb light of particular wavelengths. If you've ever seen the electromagnetic spectrum, you could go Google that, and they'll use the Greek letter lambda that looks like this to represent wavelength. And it'll have something like this, like wavelength will be on the y-axis, and they'll have, oh man, drawing a rainbow is going to be a pain in the butt here, isn't it? Let's see, I'll start with, no I won't. Blue, indigo, violet, all right, green, G-R-O-Y, I need yellow. You have seen this before, probably. Okay, so there we go, Roy G. Biv. This is visible light, and oh gosh, I forget which direction it goes, but like UV light will be up here, and X-rays will be down here, and like radio waves here. But the visible light spectrum is like from here to here in the middle of this. Some pigments will absorb light, over this range of wavelengths. But plants will have a whole different mix of wavelengths to them. I don't know if you actually did that virtual lab where you had to separate pigments. I'm not teaching the 101 lab, so I'm not 100% sure where you guys are at there. But plants will have a mix of pigments, and some of them will absorb light over this amount of wavelength, and then other pigments will absorb light over this range of wavelengths. So now they can absorb light over all that right there. So just a little tidbit there. And I'm spending time on this because we're like, oh, plants use energy from the sun and to make ATP for themselves. Like, well, how do they do that? They do that in part because pigments can absorb light of different wavelengths. Alrighty. I am not going to test you on the electromagnetic spectrum. Ooh, that was fun. Really like this eraser action here. Ha, can make more room for myself. I am not going to test you on the electromagnetic spectrum, but you do need to know that pigments are involved in this process. Okay, so we can get rid of this stuff here. Alrighty, so in the light dependent reactions, pigments will absorb what we call a photon or photons of light of particular wavelengths. I misspelled wavelengths. Oh well. A photon is just basically energy from light. So what happens is that, here, let's use yellow for sunlight. Why not? Light will be absorbed by the pigments that are inside the chloroplast. Those pigments are able to transfer that light energy over to a water molecule. So we need something we can write in here. Okay, pigments, and again, this is in the light-dependent reaction, pigments transfer this light energy eventually to the water molecule like this, and get the water molecule to split up. And get the water molecule to split up. So for example, this is what happens when a water molecule splits up, but it takes energy from light to do this. Let's see, we got to start with 2 water molecules, and we'll end up getting 4 electrons, 4 protons, and an O2 molecule. Let's do that in blue, just because. There we go. Does this look at all familiar? Because it's basically the reverse thing that happens at the end of chemiosmosis when oxygen takes on 4 protons and 4 electrons to keep that gradient going, and you put them together and you get water molecules. The opposite is happening, and it's the first thing that happens in photosynthesis in the light-dependent reactions. So, oops. Pigments transfer light energy to the water molecule. So light energy via pigments. And get this water molecule here to basically spit out 4 electrons, 4 protons, and some oxygen. So look back here at this chemical reaction here. We need a different color. This part here happens first. See, I didn't lie. It happens first, it happens right away. So we get oxygen being produced right away in the chloroplast. In cellular respiration, oxygen got involved, way the heck at the end. This is the first thing that happens in photosynthesis. So poof, we get O2 being produced right away. But let's take a look at this. Let's come back here. Okay, so we've got these two water molecules that get split up to produce 4 electrons and 4 protons. These protons get pumped across the inner membrane of the chloroplast and end up doing their funky chemiosmosis diffusion dance thing. It sets up a concentration gradient that's not stable. They flow back through, they equalize the gradient through ATP synthase, and that releases energy. ATP synthase uses that energy to put the phosphate group back onto ADP to make ATP, and the cell goes, yay! Right? So let's just summarize that right now, because we don't need to go through all that all over again. Chemiosmosis. Let's just say that these protons here are used in the chloroplast to do chemiosmosis. Are used in the chloroplast to do chemiosmosis and produce ATP. There we go. I could have said that better. They are used in the chloroplast to produce ATP through chemiosmosis. Let's just fix this. To produce ATP through chemiosmosis. There we go. So this part back here, we're done. All right. So in here in the chloroplast, let's take a look in here, folks. We just produced oxygen, and we got these four protons and 4 electrons. They do their chemiosmosis diffusion dance across the inner membrane of the chloroplast. We make a ton of ATP. That is basically the light-dependent reactions. Pigments absorb energy from light of a particular wavelength. Pigments transfer this light energy to a water molecule and get that water molecule to split up. Oxygen is produced, and these protons are used in the chloroplast to produce ATP through chemiosmosis. That's the light-independent reaction. Okay, and it depends on the plant. It depends on the condition how much is made, so I'm not going to make you memorize how much. Let's just say it makes a lot. That's what those 2 arrows are for. A lot of ATP. So let's move on to the light-independent reactions. The light-independent reactions use up a ton of ATP. Of ATP that was made in the light-dependent reactions. It's made in the light-dependent reactions to do the other part of photosynthesis back here to do this part here. Only, how do you make CO2 into C6H12O2? Don't you need some H's? The H's come from here. Okay? after those hydrogens basically do their little chemiosmosis thing, they join with CO2 to form sugar molecules. So let's write this down. The light-independent reactions use up a ton of ATP that was made in the light-dependent reactions to take CO2 and build sugar molecules. Let's say that better. They take CO2 plus the protons and electrons that came from water after chemiosmosis after chemiosmosis to form sugars. Eventually, C6H12O6 can be formed. But sometimes they're just even smaller sugars than that. But for the sake of this class, we're going to stick with that. Okay. It takes a bunch of energy to put, how many did we start with? Let's take one more look. It takes a bunch of energy to put 6 carbon dioxide molecules together to form a C6H12O6. Like, for example, if this is a CO2 molecule, we got to take 1, 2, 3, 4, 5, 6 of them, put them together with a bunch of protons and electrons to form basically one 6-carbon molecule. 4, 5, 6. That takes a lot of ATP to put 6 of these individual things together to form one C6H12O6 molecule. And all that ATP comes from the light-dependent reactions. From light-dependent reactions. So let's take one more look at this and just reiterate that plants, through one full cycle of photosynthesis, will, you'll have 6, actually, you know what? Yeah, I'll write it this way. 6 CO2 plus 6 water molecules eventually forms C6H12O6 plus 6 O2 that we love to breathe in. All right. So, but this time, this happens first. This oxidation happens first in the light-dependent reactions, yeah? And it produces a ton of ATP. After that's done, only after that's done, are these 6 CO2 molecules put together to form a sugar with the leftover hydrogens that came from water. And this is a reduction, and it happens last in the light independent reactions. Yeah. Now, in your online book, they go into more detail about the light-independent reactions, and they talk about how the light-independent reactions have something called the Calvin cycle. We are not going to go through the Calvin cycle. We are not going to talk about the big enzyme called Rubisco that's in there. But I want you to get the gist of this, of what's happening here. And I want to put in a good word for my plant friends out there. Plants are so cool. Check them out. Let's draw that plant cell, that Alodia, that brick-like cell from Alodia that you saw, like, the third lab before we started social distancing. All right, and you had, oh, there's a big central vacuole there, but you saw all these bubbly looking things that were basically, those are the chloroplasts. What you couldn't see under the microscope was that plants also have mitochondria. Lots of them. They're just smaller and you can't see them as easily as you can see the chloroplasts. Take it back. All right, now in the chloroplast, plants are doing this whole thing up here. They're doing photosynthesis, and they're taking CO2, using energy that from sunlight that got the water molecule to spit out some oxygen and electrons and protons to do chemiosmosis, and eventually build a sugar molecule. Right? So we're doing this in the chloroplast. We're getting 6 CO2 plus 6 water molecules makes a sugar molecule plus a bunch of oxygen. Yay. In the mitochondria, I mean, check it out. It just produced sugar. In the mitochondria, this sugar molecule is used to do aerobic cellular respiration, which is C6H12O6 plus 6O2, yields 6 CO2 and 6 oxygen molecules. What this means is that in the presence of sunlight, plants' chloroplasts are making a bunch of sugar molecules for themselves, right? So that later on, when there's no sunlight around, like at night, what do you do if you have the munchies at midnight and you're a plant? You get your mitochondria doing aerobic cellular respiration, where you break down that same sugar molecule, combine it with a little bit of oxygen that's in the air, probably still in your plant structure. I wrote this wrong here. This over here should be H2O. There we go. Much better. And to produce some water. Okay, what this means is that plants make their own food. That is freaking amazing. They produce sugars in the chloroplast by using energy from the sun and doing chemiosmosis that way, so that later on, when there's no sunshine around, or like when you plant a seed, when you plant a pea seed, no, you didn't do that one in lab, but when people plant pea seeds, those pea seeds break down all of their sugar. I mean, they're buried in the ground. There's no sunlight down in the soil. So they break down their sugars that they stored up through aerobic respiration so that they can get ATP and grow that way. Plants make their own food. Mammals can't do that. We can't do that. We don't make our own food. We have to eat food so that we can do aerobic cellular respiration in our mitochondria. Plants don't need to eat food, okay? They get the nutrients they need. They make their own food. Yeah, they need nutrients in the soil, but those are usually minerals and nitrogen and stuff like that, right? You don't need to actually feed a plant sugar. You don't need to feed a plant a piece of meat, unless we're talking about, Little Shop of Horrors, that musical movie, where the plant ate meat. Anyhow, that was a joke. Plants make their own food, and it really makes them interesting and fascinating organisms that they can do that. Algae do the same because they produce photosynthesis. So that is what makes plants really, really cool. Okay, so that finishes up our unit on how cells make energy. Because this has been a crazy semester and we do not have enough time to get through as much material as we normally would have under normal circumstances, we are going to be skipping ahead, and for the rest of the semester we are going to begin a section on inheritance. and basically a field that we call genetics, or rather, the genetics of inheritance. This is chapter 8 in your online book, section 8.1, 8.2, and 8.3. So if you want to take a look at that, please do take a look at that, definitely. All right, but basically we have known, humanity has known for thousands and thousands of years that we inherit traits from our parents. We have noticed that siblings tend to look alike, and we know that families will share these traits. Okay, so we know that we inherit traits from our parents. Now, first up, I want to talk about what this word traits mean. What does traits mean to you? A trait would be some sort of physical characteristic. What your earlobe looks like, the size of your eyes, the shape of your eyebrow, whether or not you have a straight hairline or if you have a widow's peak that's a pointy Dracula-like hairline like I have, actually. Okay, I've got a widow's peak. If you have a straight hairline or something like that, God, it looks like Batman and Robin or something like that. It's ridiculous. This unit, you guys are going to be able to laugh at my artwork a whole lot. Yeah. Anywho, traits are basically physical characteristics. So someone's eye color or skin color, whether or not their cells are able to make a particular protein, that is a physical characteristic, and these things are inherited. We learned much of what we know initially from a scientist named Gregor Mendel. Gregor Mendel was an Austrian monk and scientist back in, I don't know, I want to say he was doing most of his work in the 1860s, something like that. And he experimented with plants, inheritance in plants, because it's pretty easy to grow plants faster than it is to grow people and observe them, right? So doing experiments on plants is ethical. Doing experiments on humans, not so much. So he was working mostly with pea plants. Pea plants, peas that grow in a pod. If you've ever eaten snap peas, you can eat the pod, but peas come in pods like that before you pop the pod open and eat the peas inside, and they grow on a plant. Like I said, you can make fun of my artwork in this unit a lot. You'll have lots of opportunities for that. But he played around with growing pea plants, and he made many observations about inheritance. And these observations, what is that word that we know that in science is the observation? Think back to the first week of class. We had these scientific blanks versus scientific theories. You said scientific law, you got it going on. Gregor Mendel made observations, which are scientific laws, and we call these laws, these observations that he observed through his experiments, the laws of inheritance. Laws of inheritance. So these laws of inheritance, and actually the word that I think your online book is going to use is the laws of segregation. Segregation means separation. And in science, we're talking about the separation of traits. And hopefully that will become clear later on. So let's go through the laws of inheritance that Gregor Mendel talked about and arrived at with his experiments through pea plants here. First up, he determined that each individual has 2 factors for each trait. Each individual has 2 factors per trait. Like if your earlobe looks a particular way, that's because 2 factors determined that. But he made the observation that these factors separate during formation of the gametes. Separate during formation of the gametes. Don't know what the word gametes is? Gametes refers to sex cells, sperm and ova. Sperm and ova. Ova is a more appropriate word than egg. Sperm and egg, sperm and ova. Think of it coming from the word ovary. Here we go. So each individual has 2 factors for each trait, and these factors separate from each other during formation of the sperm and ova. Okay, so each gamete, sperm or ova, contains only 1 factor per pair of these factors. Each gamete, sperm or ova, contains only 1 factor per trait. And then finally 4, fertilization, the sperm getting together with the ova, gives the new individual 2 factors for each trait. Sperm getting together with the ova gives the new individual 2 factors per trait. Now let's talk a little bit about this. Here we go. So each individual has 2 factors per trait. Let's draw a cell and say, they've got 2 factors per trait. I'm just drawing BS here. I'm just drawing something to give us a visual. But that these factors separate from each other during formation of the gametes, the sperm and the ova, and that each gamete, each sperm and each ova contains only 1 factor per trait. So let's draw a sperm, and let's say it has only 1 of these traits, and let's draw an ova. You've seen an ova look like this before, and it's only got 1 of these traits. And that fertilization, the sperm getting together with the ova, sperm plus ova, if we put these 2 together, give the new individual, oh my gosh, these look like eyeballs and this looks like a nose. See, you get to make all kinds of fun of my artwork. The sperm here getting together with the ova gives the new individual 2 factors per trait. 1, 2, like that. We're going to come back and talk about this a little bit more. So, make sure you can find this in your notes. And all right, so let's take a look at what Mendel did and how he figured out some of these things. One thing that he noticed that actually kind of got him started in genetics and in studying inheritance was that he noticed that some traits in plants showed up more often than others. Some physical characteristics, some traits showed up more often. So for example, he found that it was more common for pea plants to be tall. it was more common for pea plants to have a tall height than for pea plants to be short. See what I mean? You can make all kinds of fun of my drawing here. So I was like, you know what I'm going to do? It's more common for plants to be tall than short. I'm going to breed these two plants together. Now, I am not a plant breeder. I know that it has something to do with taking pollen from the flower of one plant and putting it onto the flower of another plant and then collecting those seeds later and planting those seeds and seeing what the plant babies look like. Something like that. But when we show that we bred two plants to each other, we basically just put an X little between them. So this little X here, sometimes it's a cross. It means that we bred them together. So he took a tall pea plant and bred it with a short pea plant and then checked out what the baby plants looked like. All right, so this parental generation here, we call that the pea generation. All right. I'm going to draw a line showing their plant babies. They found that all these plant babies were tall. All tall. 100% tall. This is actually called the first generation, or the F1 generation. And everybody in the F1 generation was freaking tall. No big whoop. I would have stopped. I would have been like, fine, tall is more common. It dominates over short. No big whoop. But Mendel was much smarter than I am. And he's like, maybe for hoots and giggles, I'm going to take two of these F1 generation plants here and here, and I'm going to breed two of them together. And I'm going to see what's going to happen. All right, so he did that and looked at the plant babies, bred those two together, and he got a bunch of tall plants. Surprise, surprise, I would be like, see, told you so. But he'd be like, I'm smarter. Because 1 in every 4 plant turned out to be short. So he had 75% tall, 25%, or 1 in 4, would be short. And that made him go, there is something in this short plant here that came from this generation up here, from the grandparents, from the P generation, that was still present in the F1 generation. It was still present in here, but it didn't show up until later. It was experiments like this that led him to call traits certain things and say, oh, the one that shows up more often, the tallness, that is dominant. And it's hiding the expression of the shortness here. And it didn't come out until this next generation up, which would be called the F2 generation. So it's like, okay, we need some new vocabulary here because nobody's ever talked about this stuff before. So some traits that show up more often, he called these dominant traits. And the other ones he called, you know what this word is, probably some of you, recessive. Dominant traits and recessive. Dominant traits hide or mask expression of the recessive trait right there. So he's like, all right, well, this is weird because both this dominant form, there's some dominant factor here that causes tallness, but it's got a different version that causes not tallness, that causes a lack of tallness, shortness. That there are somehow two factors affecting and controlling the same trait. We use a different word for that now. He used the word factor. We use factors up here. Factors. Factor, or whatever the heck the German word for factor is. I don't know that. But we use a different word. And if you're thinking we use the word genes, you're almost there. We use a different word that I bet some of you have heard before, and it's called an allele. Alleles are alternate forms of the same gene. And I gave you a definition for the gene much earlier in the year. So just to refresh our memories, remember a gene is the basic unit of inheritance. Unit of inheritance. But it also is an area of DNA that contains information to build a protein. This is just review here. So now I'm telling you, the same gene can have slightly different forms, and we call those alleles. So instead of the word factor here, let's back up here. Each individual has 2 alleles for the same trait. These alleles separate from each other during formation of the sperm and ova. And each sperm and ova contains 1 allele per trait. And fertilization gives the new individual 2 alleles per trait. Right. So alleles are alternate forms of the same gene, and they can be dominant or recessive. So let's write this down here. So dominant alleles. They hide or mask what we call expression of a recessive allele. Dominant alleles get a capital letter when we write this stuff out. Like for this tallness, if tall is dominant over shortness, tall is the dominant allele, short's the recessive allele, I would give tallness a capital T. Recessive alleles get a lowercase letter, a lowercase version of the same letter. Let's say it that way. Like, I'm not going to give it an S for short. I'm going to give it a T, but I'm going to give it a lowercase t. Right there. And we will pick up where we left off next time. Bye-bye. So we have both dominant alleles and recessive alleles, and the presence of a dominant allele hides expression of the recessive. Now let's think about what this means in terms of the laws of inheritance. So I'm just going to scoot back here and back up to Mendel's laws of inheritance. Each individual has 2 alleles per trait. So in terms of tallness or lack of tallness in pea plants, let's take a look at the different combinations. So let's see. Every individual has 2 alleles per trait. And if we're talking about an individual plant cell, each plant is going to have 2 letter T's in its cells, right? So let's look at combinations. Combinations of alleles. Actually, let's do this a little differently. We'll make a little bit of a table, why not? Like, let's say, okay, a plant could have 2 dominant alleles. Right there. Actually, let me move these. I'm going to give myself a little more room here. Here we go. A plant could have 2 dominant alleles, so 2 capital T's here. Or plant could have two recessive alleles, so two lowercase t's. Why not? Is there another option? Why, yes, there is another option, one of each. A plant could have a dominant allele and a recessive allele, so I'll write this as a capital T and a lowercase t. There we go. All right. So let's talk about what each of these scenarios, what each plant is going to look like in this case. So if a plant has two dominant alleles, up here is where we are, what's this going to look like? It's going to be a tall plant, all right? Like this one up here, the original one, this plant here. Let's say it has two capital T's. It's a tall plant. Because tallness is caused by that capital T allele, that dominant allele. How about these two lowercase t's? What's that plant going to look like? It's going to be a short plant. Miss getting to see everybody and hear you and have a conversation about this. We're doing what we can here. Somebody out there, I can feel it, said that was a short plant. Two lowercase t's, two recessive alleles, you get a short pea plant. Oh, let's even draw one. Let's draw a little short plant. And here, I'll draw a tall one here. There we go. But what about this one down here? What's this plant going to look like? It's got one dominant allele that says the plant's going to be tall, and it's got one recessive allele that says that the plant's going to be short. But the way this whole dominant recessive thing works is that this dominant allele hides, it overpowers the presence of a recessive allele. And so this plant is also going to be tall. Make it tall. There we go. Now we need some words here. Let's get a different color for these words. Let's go, pick a color I haven't used in a while. Get rid of this. There we go. Here, this side here, these things, the different combinations of alleles that a plant could have, we give a name to. This is called the plant's genotype. Genotype. The genotype is the combination of alleles, what the alleles are. Let's make a little room there. What the alleles are. And remember, alleles are just alternate forms of the same gene. This dominant allele causes height, or height in plant, affects the height of a plant, and this recessive allele affects the height of the plant too, just in different ways. Right, so these combinations of alleles is called the genotype. I know some of you have heard these words before. What's the other one? What are these things here? What the trait actually looks like, or what the plant looks like? Not genotype, it's ding ding, phenotype. This is called phenotype. This is what the trait actually looks like. So, and here's one way to remember it. Genotype over here has the word gene in it. So it's like what's going on with the genes, what's going on with the alleles. Phenotype begins with a PH, which also starts off the word physical. It's what the physical trait looks like. Tall plant or short plant, what the plant looks like, what the physical manifestation is. So we got to keep our genotype straight from our phenotype right there. Okay, so here's a question for us all. Let's get a third color, shall we? All right, can you look at, or rather, not look, can you know a genotype and determine the phenotype? If you know a genotype, can you determine the phenotype? Sure, of course you can. That's what we just did here, right? We're like, okay, 2 capital T's. That means it's gonna be a tall plant. We use genotype, 2 capital T's, 2 dominant alleles just to know that that would be a tall plant. So sure, we can do that, for sure. But how about this? Let's switch this up just a little bit. And how about, can you know a phenotype and determine the genotype? Can you know phenotype up here? Can you look at the phenotype, whether a plant is tall or short, and know what its genotype is? Think about that for a sec. Some of you are saying yes, thinking yes, you can know a phenotype and determine its genotype, and some of you are saying no, and some of you are saying, which is the correct answer, some of you are saying, sometimes. Sometimes you can look at the phenotype and know what the genotype is. For example, you can look at a short plant right here, and you know that a short plant's going to be 2 recessive alleles, lowercase t, lowercase t. But can you look at a tall plant and know for sure what its genotype is? And in this scenario, no, you can't look at this tall plant or you can't look at this tall plant or this tall plant and say, oh, I'm looking at this tall plant right here, and I can see for sure that it's got 1 dominant allele and 1 recessive allele. No, you can't. You cannot look at a tall plant, tall pea plant in this case, and say, is it capital T, 2 dominant alleles, or a dominant allele and a recessive allele, capital T, lowercase t? We don't know by looking at the plant. So the answer to this question here is sometimes. But that's because we have 2 options for tall, right? We've got this and this. So what this means is that we need more words to describe these things. We need more words. Let's clean this up just a little bit. Oops. There we go. Because do you see how this genotype here with a dominant allele and a dominant allele, 2 dominant alleles are capital T, 2 of the same alleles there? Do we have this scenario in any of these other genotypes here at all? Here's where, if we were in class, well, we are in class, what the heck, I'll do it anyway. On Sesame Street, how they used to, maybe they still do, I haven't seen Sesame Street in about 10 years. How they used to play this game, they would have 3 things out and they would sing the stupid little song going, one of these things is not like the other, one of these things is not the same, or something like that. I don't know how the song goes anyhow. Sorry to subject you to that. And people would be like, oh, well, there's a red ball there and it doesn't fit in with all the other, squares that are there or something like that. But hang with me here for just a second. What I'm saying is that these 2 of these 3 genotypes have something in common. And what I'm getting at is that this one here, right here, has 2 of the same alleles, 2 dominant alleles. And this one here also has 2 of the same allele, only it has 2 recessive alleles in that case. We say that these 2 genotypes, and let me get rid of these circles, we say that these are what we call homozygous. Homozygous. It means having two of the same alleles. Having two of the same alleles. But this one here, that's not homozygous. It has two different alleles. It has one of each. So it's not homozygous. We call it, what is it? Somebody's got it. And somebody's going, oh, that is heterozygous. Heterozygous. Homozygous means having two of the same alleles. Heterozygous means having two different alleles. Different alleles. And for those who are curious, this zygous part refers to like zygote, which is a stage of a fertilized embryo, so a developing embryo. So same root word there. All right, so we are getting lots and lots of vocabulary here, aren't we? We have dominant and recessive. We have genotype and phenotype. We got to keep those straight. We have homozygous and heterozygous. And from now on, we are not going to be using words like big T, little t, little t. No, we're going to combine them and say words like, oh, heterozygous, to mean one of each allele. But here's the rub. If I say something, oh, well, this plant is homozygous, well, which one is it? Is it homozygous for the dominant allele or homozygous for the recessive allele? So we have to use our words. We have to use words like homozygous recessive. And in this case, it would mean two recessive alleles, and I would draw it as two lowercase t's. But don't be saying little t, or for homozygous dominant. That would be two capital T's. But don't be saying big T, little t, little t. That's starting to sound like a Dr. Seuss book. Big T, what begins with T? I see a tufted turtle looking at me, or something like that. No, we are going to use science words, homozygous recessive, homozygous dominant, or heterozygous, which is one of each. So you don't have to say heterozygous dominant or heterozygous recessive. There's no such thing there. You just say heterozygous because it means you have one of each. So we're going to be using fancy science words with that. Okay, so we got homozygous recessive, homozygous dominant, heterozygous, genotype, phenotype. We got to keep all that stuff straight. Let's start to use these. Let's start to use these words for some genetics problems and looking at what's going on. But let's think back to Darwin was actually doing science around the same time as Gregor Mendel, but they didn't really know about each other's work. But we're going to go back to Gregor Mendel's experiment, where he took a tall pea plant and bred it with a short one. And all the babies in the F1 generation here were 100% tall. Let's do this experiment in terms of genotype. All right, so I'll draw this again so we don't have to be scrolling back and I don't have to make everybody's head spin from my moving the screen around so much. But let's look at Gregor Mendel's original experiment. A tall plant crossed with a short one. The tall plant that he started off with, it was homozygous dominant, so I'm going to write two dominant alleles there. And the short plant, what's its genotype going to be? What do I draw for T's here? Two lowercase t's. That is the only option for a genotype for a short pea plant here. So they crossed them together. All right, now what this means is that this tall plant here can donate through its plant gametes, and gametes are sex cells. All right, it can donate to its plant babies in its gametes either this allele or this allele. But in this case, they're both the same, right? So when all of these babies were born, they got either this dominant allele or this dominant allele. But either way, this tall pea plant here can only donate a dominant allele to its plant babies. So it can only give its babies a dominant allele. Likewise, this short plant in its plant gametes can either donate this allele or this allele to its plant babies. What's the difference? They're the same. It can only donate a recessive allele. So 100% of their plant babies were tall. If you think back. Oh boy, I'm getting really crappy with my pea plant drawing here, right? So they were 100% tall, and their genotype all had to be heterozygous. 100% tall and heterozygous. That's an easy one to work out because this tall plant over here could only donate a dominant allele. Can only give its babies, its plant babies, a dominant allele. Whereas the short plant over here can only give its plant babies a recessive allele. Yeah. Things get a little more complex when Mendel bred two of these heterozygous plants together. In this case, let me move this here. Get that out of there. When these two are crossed, this plant here, this one here, I'll just color it in purple. Gee, we have a purple pea plant now. Can either donate this dominant allele to its babies, or it could donate this recessive allele to its babies. And this one over here that I'll color in purple now, just so you can see what I'm pointing to, can either donate a dominant allele to its babies or this recessive allele to its babies. Like that. So it's not as clear cut. We need a way to figure this out, and some of you have already seen this before. Some of you probably remember genetics from another class. So what we need to do is do something called a Punnett square. So we need a Punnett square. And by Punnett square, I mean we're literally drawing a square box. A square, like that. And we're going to turn it into a 4 square box. You know, like those 4 square game that you see on playgrounds, where 4 people play and there's 1 person in each of the squares. So we're going to divide each of these in half. And what we're going to do is up here, we're going to put the possibilities for different alleles that one plant can donate here at the top of each column. And then we'll take the other parent plant and put the possibilities for alleles that they can give at the end of each row. That was a crappy thing. There we go. At the end of each row here will be the possibilities of T's that one parent can give, and at the top of each column will be the possibilities that another parent can give. So let me just erase that so we can get started with it. Okay, let's get back to green. All right, so I'll take this parent right here. Right up there is where we're looking. They can donate either a capital T to its plant baby, so I'll put the capital T up there. Or it could donate this lowercase t to its babies, so I'll put the lowercase t at the top of the second column. Now let's take this plant right here. It can either donate this capital T, this dominant allele to its babies, I'll put it at the end of the first row, or it can donate a recessive allele to its plant babies. So I'll put this recessive allele at the end of the second row. And think of it this way. Think of it, oops, what did I do there? And how do I get rid of it? There we go. Think of it this way, that what I put up here, these capital T's and this dominant allele and this recessive allele here, these are like the plant sperm. I don't know my plant biology very well. What the male gametes are called, or those gymnosperms, I don't know. So think of it that way. And think of these ones over here at the end of these column as kind of the ova, if you want. That sometimes works. Because the two factors for each, the two alleles for each trait separate from each other upon formation of the gamete, and some sperm could have a recessive allele in them, and some could have a dominant allele in them. So if that helps. Meanwhile, this is looking a little messy here. There we go. So what we're going to do is we're going to fill in, oops, I did not mean to do that. I have to go back to my pen first. We need to fill in these boxes, don't we? You don't have to color in your box like that. I just wanted to show you that what I'm putting in this box here in the upper left is that I'll grab the allele that's at the top of the column here, put it right in there, and grab the one from the end of the row that was right there and pop it in there. Clean that up. All right. For this box right here, I'm going to take the T that's at the top of the column, put it in there, and put the allele that's at the end of the row in there too. Notice that I put the capital letter first. We're not going to write it like that. No. In genetics, we put the dominant allele first. So only I things do we really, like iPad, which is what I'm writing on right now, do we use a lowercase. Why am I doing that? All right, so in here we're going to have a capital T because that's what's at the top of the column, and a lowercase t from the end of the row. In here, top of the column, there's a recessive allele. End of the row, there is a recessive allele as well. So there we go. All right, so we got 4 possibilities for their babies here. And I'm just coloring these in so that you can see what I'm pointing to. All right, here's what I'm looking at. Out of 4 boxes, did it again. Out of 4 boxes, this plant, that's going to be a tall plant, right? 2 dominant alleles, tall. Here it's heterozygous. That plant's going to be tall. That's going to this plant here is going to be tall. This one will be short. So 3 boxes out of 4 total boxes are tall. So there's a 3 in 4 chance of a tall plant with this cross. You can divide it out and say 75%, that's fine, or you can just leave it as 3 out of 4, that's fine. But only one box out of 4 is short. There we go. Now that's if I'm asking you for phenotype. We're talking phenotype here, aren't we? About whether or not a plant is tall or short. But what if I asked you a different question? What if I asked you, what is the likelihood that this cross results in offspring that have the same genotype as the parents? So what are we looking for if we're talking genotype? We're looking for, okay, the parents up here, let me just erase those little circles. The parents were both heterozygous, right? We basically crossed a heterozygous plant with another heterozygous plant. And we put one capital T and one lowercase t here, and one dominant allele, one lowercase recessive allele here. So what's the likelihood of that? So you go fish. You go looking for the parent's genotype, and I'm going, this one's heterozygous, this one's heterozygous. 2 boxes out of 4. That reduces to 1 in 2, or 50% chance. So you got to be clear about what I'm asking you for, right? You got to be clear. Am I asking you about phenotype? You know, what's the likelihood of having a tall plant with this cross? Well, it's 3 in 4. What's the likelihood of having a short plant? 1 in 4. What's the likelihood of their having the same genotype as the parents? 1 in 2, or 2 out of 4, whatever. How about what is the chance of their offspring being homozygous dominant? Homozygous dominant. Right here. Only 1. 1 in 4. So it's 1 in 4, or, 25%. Whatever trips your trigger. You can give me the fraction, or you can divide it out. Fractions are fine with me. They're easy. Works for me. Alrighty. So let's see. That's tallness or lack thereof in plants. Let's do something human. Want to feel human. Now let's look at another trait. Let's look at a trait in humans. Let's get a different color here. Getting really bored with these. Gosh, I'm so indecisive. Nah, something a little brighter. Yeah, let's go with this purple. Why not? Let's look at earlobes in humans. Earlobes in humans. Oh, you know what? I think I do a different color. Earlobes in humans. There are different shaped earlobes in humans. For example, a dominant allele, and I'll use the letter E because E is for earlobes. If an individual with a dominant earlobe allele will have an earlobe that looks like, oh gosh, maybe make a finer point here. We'll have an earlobe that looks like that. Please do not laugh. You know it looks like a fish. This is what I call a dangly earlobe, or sometimes it's called a free earlobe, or I've even seen it called unattached, meaning it's dangly down there at the bottom of the lobe, right? I mean, I know everybody's earlobe is attached, but whatever. But a recessive earlobe allele will give you an earlobe that looks like mine. My earlobes look like that. Yes, I know it looks like a fish. Let's see. I should have chosen orange. This could have been Nemo. Yeah, let's. Here we go. It's a clownfish. All right, this is called an attached earlobe. An attached lobe. Can you see the difference? This attached earlobe down here, it's attached lower. People with free earlobes that look like that, I mean, that is like the worst earlobe I've ever drawn in my life. They'll have like, I don't know, it has more of a curve to it. And I mean, I don't know, go look in the mirror and check out your earlobe. Check out the earlobes of the people that you might be living with. And I don't know, take a poll, because I would be taking a poll right now if we were having class in person. And usually there are usually more people in class that have that dangly earlobe than have attached. All right, so let's make a little room here. There we go. What are the possibilities for people with dangly earlobes? What are the possible genotypes? What are their genotypes? Like, for example, people with a dangly earlobe, and yes, I know that's just a stupid word for it, but it amuses some people, so it amuses me. Why not? And I think it's easier to say than attached and unattached, because it's not unattached. Your earlobe is attached at some point. But people with dangly earlobes could be homozygous dominant, right? Or they could be heterozygous, because this dominant earlobe allele completely dominates and hides expression of the recessive. But the attached earlobe here, earlobe that I have, there's only one possibility there. It's got to be homozygous recessive. Now don't be going little e, okay? We have to use fancy science words. This is, can't write. This is homozygous recessive. Only one possibility for people who have attached earlobes. They are homozygous recessive. So I know this is me. This is me. Here we go. So here's an interesting proposition. Here's an interesting puzzle. Okay, if this is me, this is Mary, right there. I have, let's see, Mary has attached earlobes. She has a son with dangly earlobes. What does this tell us? What does this tell us? Like if I draw Mary right here, I know I look like a fish. We know that I am homozygous recessive, but I've got a kid with dangly earlobes. All right, do we know my son's genotype? Do we know his genotype? Yes, we do. We know his genotype. Okay, because look up here. These are the only two possibilities for having dangly earlobes, homozygous dominant or homozygous recessive, and there is no way my son could be homozygous dominant because I only have a recessive allele to give him. So I gave him a recessive allele, which means he's got to have, if he's got dangly earlobes, he's got to have a dominant allele present as well. And folks, where did my kid get this dominant allele? Because it didn't get it from me. He had to have gotten it from his birth father, right? Okay, so this had to come from his dad. His dad, right? So his dad had to have at least had one dominant earlobe allele. So what kind of earlobes did his dad have? Does his dad have? He's got dangly lobes. Dangly lobes. The dad had dangly lobes. So just because I'm into drawing Stupid looking earlobes. There we go. All right. Do we know what his dad's other allele was? Do we know? Could it have been recessive? Sure. Could it have been dominant? Sure. We don't know. Or do we? What would tell us what his other allele is? How could we know? Think about that for a sec. We could look at his parents. We could look at his parents. In-laws. Yeah. They're not around anymore, though. All right. But if one of his parents had attached earlobes like this, then we would know that he had a parent who had those, and he would be heterozygous, sure. But if they're not around, is there any other way we might know? Well, we might know if I had another kid. If we had another kid. Here's kid 1. And yes, we do have kid 2. And it turns out that kid 2 has attached lobes. So what does that tell us what this kid's genotype is if he has attached lobes? Yes, you got it right. He's got to be homozygous recessive. Sure, he probably got one of these. Well, I know he got one of those recessive alleles from me, but the other one he had to get from his father. So his dad must be, capital E, lowercase e, heterozygous. Right? So in this problem, we kind of had to think backwards, didn't we? We had to think back to the parents. When you see someone, like let's look at kid 1 right here. When you see someone, dangly lobes, say he's heterozygous in this case, we get one allele from one parent and one allele from the other. Like this allele could be from this person's, some unknown person, 1, whatever. Could be from their mom or dad. Or this one could be from their mom or dad. But I guess what I'm saying here is this allele could be from their mom and this one could be from their birth dad, or this allele could be from their mom and this one be from their birth dad. Either way, you get one allele from one parent and one allele from the other. So rather, let's say we inherit one allele from one parent and one allele from the other. We inherit, I could have said this more simply, we inherit one allele from our birth mothers, we inherit the other allele from our birth fathers. That's how it works. That's how sexual reproduction and inheritance through sexual reproduction works. We inherit one allele from our birth mother's ova, her gametes, and we inherit our other allele from our birth father's gamete, our birth father's sperm. We just don't necessarily know which is which sometimes. There we go. So there is that. Let's see what else can we come up with. Earlobes. Actually, check out your earlobes and see if you can figure out if you have dangly lobes or attached lobes. Why not? Oh, you know what? Let's do this problem here. Let's say, let's go back to this. You know what? No, we haven't done brown in a while. Let's say a man with attached earlobes has children with a female, I'll use the universal symbol for female right there, with a female who is heterozygous for the trait. What is the probability they will have children who are homozygous recessive? So there I'm asking you for genotype there. Here's one way to approach this. Write down everything you know that we can figure out from each sentence here. So, a man with attached earlobes, what does that tell us? Is attached earlobes, that means his earlobe's gonna be like that. So we know this guy's gotta be homozygous recessive. So the man is homozygous recessive. But he's having children with a woman, with a female who is heterozygous. So she is heterozygous for the trait. Let's set up this cross. We are crossing a male who is homozygous recessive. Don't say little e, okay. And having children with a female who is heterozygous. All right, let's set up our Punnett square. This is actually a pretty basic Punnett square, isn't it? There we go. Let's take this parent over here and say, okay, she can donate a dominant allele to her offspring, or she could donate a recessive allele to her offspring. Let's come over here and do this parent. This guy can donate a recessive allele, or could donate a recessive allele. So we don't really need a full box for this, but I just wanted to put it together just so that we get used to doing a full box. I feel like I'm one Fruit Loop short of a full box. No, that was just a silly little saying. Okay, so let's fill in the boxes. Capital E at the top of this one here goes in here. This one here. Whoop. End up with this. Here we go. Here we go. So, what did we ask here? What is the probability that they will have children who are homozygous recessive? So we're looking for homozygous recessive, which is 2 lowercase e's, 2 recessive alleles. This one out of 4, so 2 out of 4. 2 out of 4, or, 50% chance. Yeah. What if I asked a different question? What if I wanted to know what is the probability that they will have children who have dangly earlobes? There we go. Dangly earlobes. This one's dangly, this one's dangly. 2 and 4, or again, a 50% chance. Is there any chance of their having a homozygous dominant child? Nope, because this parent right here only has recessive alleles to give. There we go. All right, so these are pretty basic little problems here that we come up with. You just need to make sure you know all these different vocabularies. You need to know genotype versus phenotype. You need to know homozygous versus heterozygous. And if we're talking homozygous, we need to be able to say homozygous dominant, or homozygous recessive. And know that everyone has 2 alleles per trait. Every organism, every plant, everyone has 2 alleles per trait. one allele came from your birth mother, birth mom, and one allele came from your birth father, your birth dad. There we go. So if we're talking any random trait, like, if I say something like, someone is heterozygous for the W gene. I don't know when the W gene actually controls whether or not, actually it does. It controls whether or not you have a widow's peak that I talked about before. Someone whose hairline kind of goes like that. Looks like they're wearing a superhero suit or something. That is called a widow's peak, whereas someone who has a straight hairline looks like they have bangs. Sheesh. Okay, this is just looking absolutely ridiculous. But hopefully, go look at your hairline, people. Some people have a little pointy hairline like this. That's called a widow's peak. And this lowercase one is a straight hairline. I don't know. If we look at this and say someone's heterozygous, okay, they're going to have a widow's peak, right? This one had to come from one parent, and this one had to come from another parent. We inherit one from our birth mother, one from our birth father. We just don't necessarily know which is which unless we know a little bit more about what the birth parents' hairlines look like, that sort of thing. But when you have children, when you have offspring, you can donate either this to your kids or this to your kids. One or the other. You don't give both. You give one or the other. So if you're heterozygous for something, you could give either a dominant allele or a recessive allele to your kids. All right. Let's look at another scenario. Let's leave humans for a little bit, shall we? Let's go back to plants for just a minute. Okay, there is a flowering plant called 4 o'clocks. I don't know why they're called 4 o'clocks. My best guess would be that the flowers on this particular plant open later in the day. Kind of like morning glories are called morning glories because they bloom in the morning, and by late afternoon, that bloom has faded. It shrivels up. And maybe 4 o'clocks just open late in the day. I don't know. There is a dominant allele that causes red flowers. So I'm going to give the dominant allele a capital R, R for red, and a capital letter because it's dominant. So causes 4 o'clocks to have red flowers. There we go. I can draw a simple flower much better than I can draw an earlobe. There we go. There is therefore a recessive allele for the same gene. Alleles are different versions of the same gene, and this R gene affects the colors of the flowers. So there's a recessive allele, so we'll give it a lowercase r. All right. And it causes the flowers to be white. How do I draw a white flower? There we go. It's as white as we're going to get on a white background. All right, so what if I cross a red flowering 4 o'clock that is homozygous dominant? So it's homozygous dominant. And I'm going to cross it with a white flowering plant. What is the genotype of a white flowering 4 o'clock? It's got to be homozygous recessive, right? So this is the cross we're doing right here. We don't need to draw a Punnett square, do we? At all, because this plant here can only donate a dominant allele. It could donate this dominant allele or this dominant allele, but they're both dominant alleles, so it can only give its plant babies a dominant allele. And likewise, this one over here can either donate this recessive allele to its babies or this recessive allele to its babies. So either way, its plant babies are only going to get a recessive allele from this plant over here. Right? So 100% of the offspring, 100% will be heterozygous. What color flower will they have? Wrong. They will not have red flowers. Ha ha ha. Instead, they're going to have, because I tricked you, they are going to be pink. Pink flowers. That is a terrible looking pink flower. Let me try that again. That's a little bit better. And before you get all what the fudge on me here, going, wait a minute, I don't understand. This whole dominant recessive thing, shouldn't this dominant R dominate over this one and they should all be red? But they're not, because this, I tricked you, is an example of what we call incomplete dominance. Incomplete dominance. In incomplete dominance, the dominant allele does not completely dominate expression of the recessive. So we get a little bit of whiteness mixing with a little bit of redness, and when you mix red with white, you know that you get pink, right? This is incomplete dominance. So let's write this down. The dominant allele doesn't completely mask or hide expression of the recessive allele. So you get a blending of traits, and when you mix red with white, you get pink. Yeah, pretty cool. Now way back here, with all this other stuff, with the earlobe stuff and with the plant tallness or not, these are complete dominance. That's complete dominance. So what I would like you to do, I will tell you if it's an incomplete dominance, if it's an example of incomplete dominance. Otherwise, if I don't tell you, assume that it is complete dominance. So, assume complete dominance unless I tell you otherwise. If we didn't have to be social distancing right now, there would have been a really cool lab for you to do. That's a cat genetics lab. I'm still trying to work out if there's even a possibility that I could do it with you in class, but this way. But yeah, it's really more of a hands-on activity thing, but there are examples of incomplete dominance in that lab, and I'll give you a few more examples of them as we go on. So I will tell you. Otherwise, you can assume complete dominance, and we'll see a few more examples of that later, but this is enough for this week. I decided to try things a little differently this time by just putting up both lectures for you in one video. So I mean, it just saves me one upload right there, and you can stop it and bookmark it, do whatever works for you, and take notes. But I certainly don't expect you to sit through 2 hours of all this stuff. But yeah, so this is it for the week. I am preparing a homework assignment for you, and I will send you an email about that as well. So please be looking on Moodle and keeping up with things there. Please be keeping up with your lab stuff as well, because we are in the home stretch of the semester. So after this week, we just have one more full week and then a little cleanup, and we will be done. I will talk about the final at another time once we've decided on how this is going to all work. And I hope you have a great week. Bye-bye.